Welcome to Chapter 13, where we're going to have the Tca cycle at the heart of our coverage. This cycle, also known as the Krebs cycle or the citric acid cycle, the cycle of many names. But really, this cycle should actually be given the name stripping and ripping of high-energy electrons to produce more reducing power. Because when we talk about the Tca cycle, what we're really talking about is the further oxidation fate of pyruvate. So I hope that everyone has found the perfect medium on which to make your poster, and you're beginning to develop a very detailed poster, much more detailed than what I can draw on my computer screen, but something that is serving to allow you to follow the course of a glucose molecule as it's getting stripped and ripped of its high energy electrons. When we look at pyruvate being the product of glycolysis and our previous coverage, our now focus wants to be on pyruvate getting further oxidized. And in the eukaryotic cell, we know that the fate further oxidation occurs within the mitochondrial matrix. So the very first thing that has to happen to pyruvate before it can be further oxidized is that it is taken into the mitochondrial matrix, where these pyruvate molecules are indeed stripped and ripped of more high energy electrons, making more reducing power. And we're seeing molecules generated such as NADH and FADH2, remember, carrying around those wicked high energy electrons and taking them eventually to the electron transport chain. So acetyl-CoA is the starting molecule for the Tca cycle. So as soon as I get my NAD pluses copacetically written down, we're going to follow our acetyl-CoA into the Tca cycle, where it will get ripped and stripped of its last meaningful electrons. So as I said, the Tca cycle really should be given the name reducing power, or site of the last ripping and stripping of electrons, because these acetyl-CoAs go into the Tca, and when the Tca turns, the last of the high energy electrons are formed in the form of reducing power. So the interesting thing about the Tca cycle is that we could call it the hub of aerobic metabolism. But it's interesting to note that nowhere in our journey through Tca will we actually see oxygen used. The reason that it's called the hub of aerobic metabolism is because it makes so darn much reducing power that we're going to see that reducing power needing to have an electron transport chain and a terminal electron acceptor like oxygen in order to run. So the Tca quite simply can't run if the ETC can't run, because it makes so much reducing power. And that's a big reason it's called the hub of aerobic metabolism. But we can also think of the Tca as a hub for another reason. That is that it is kind of this spot where we can see more intermediates go in or out, and at any given time, these intermediates in the Tca that we see at each one of these reactions can either get withdrawn or input. We can have Tca fill-up reactions, and those can create a faster cycle. So if the Tca gets more of this intermediate or this intermediate or this intermediate, it'll make the Tca go faster. So we can see the Tca very much ebbing and flowing and fluxing, and very much being the hub of aerobic metabolism. The other cool thing about the Tca cycle is that the cycle itself acts almost like a catalyst, because despite the fact that an acetyl-CoA can come in and get completely oxidized, the intermediates constantly stay the same, and it's like the cycle itself is unchanged by the fact that it keeps cycling. So we sometimes refer to Tca as being like a catalyst in that particular way, because the cycle stays the same even though it's constantly turning and pumping out, new products and bringing in new reactants. So I want to write down a reaction that maybe is like the bare bones of everything that has been happening in our metabolism so far. And you all are going to recognize this reaction where we just have C6H12O6. Oh man, right away you recognize that as a monosaccharide, right? Plus O2 is going to yield 6 CO2s plus 6 water. Oh yeah, some of you chemists, are looking at the screen and you're like, yeah, that's a sweet combustion. That's throwing the jelly donut onto the bonfire and watching it go up in flames. Yes, it is. It is a combustion. But notice that also, if we think about this in terms of metabolism, this is exactly what's been happening to our glucose. This is glucose. This is glucose getting oxidized to CO2 and water. And in fact, as we see the process of further oxidation finish off, not only is acetyl-CoA getting ripped and stripped of its high energy electrons and pyruvate ripped and stripped of its high energy electrons, but we're giving off thoroughly worn out, fully oxidized, totally used up CO2. There's some CO2 lost here, there's some CO2 lost here, and then CO2 lost here. And so we're seeing these products of the combustion of glucose in a controlled way within the cellular environment. Notice too, if you want to see that other product of full, complete oxidation or catabolism or combustion of glucose, notice that the H2O, right? We breathe out those two, CO2 and H2O, fully worn out, thoroughly oxidized CO2, and notice that when oxygen is reduced at the ETC, the electron transport chain, even though this is really little and hard to see, I'm going to rewrite it, H2O is the other product of aerobic metabolism. So, although the TCA doesn't require oxygen itself, it does, in fact, produce so darn much reducing power. And as we look at the details and the nuances of these reactions, we're going to see where all that reducing power is coming from. And we're going to see that it will only continue to run when it has an ETC to which it can take that reducing power. So without further ado, we have to just dissect this, right? We're going to take this apart one piece at a time and notice that the first piece of the further oxidation fate is getting pyruvate into the mitochondrial matrix. That means having to cross two barriers. The first barrier, not very selective, but the second one, this one is a little bit more selective. So let's go back to our notes and look at how it is that pyruvate gets in to the mitochondrial matrix. So here we have it back in the cytosol where glycolysis was taking place. We recall that the yield of glycolysis was 2 NADHs and 2 ATP. And this is like the snapshot down and dirty quick and over version of glycolysis, where we had our pyruvates now made, and if their fate is further oxidation, they're going to pass first through the outer mitochondrial membrane. Now it turns out, guess what? The outer mitochondrial membrane is very much like the outer membrane of a bacterial cell. That is, it is spanned by porin proteins, and remember that those porins basically act like a sieve, allowing anything with a reasonable molecular weight to come on in. If it's traveling down its concentration gradient, it's just going to swoosh in that direction, and these porin proteins are going to allow pyruvate to enter into the inner membrane space, and then once in that inner membrane space, it's going to have to pass the more selective barrier next. So we know those porin proteins allow for free diffusion of molecules with a molecular weight of 10,000 Daltons or less. So through the porin protein into the inner membrane space, there goes pyruvate. Now, in order to get into the matrix of the mitochondrion, this is going to take a little more doing. Notice that the pyruvate in this case has to ride the PMF. That is, it has to utilize the proton gradient in order to enter into the inner membrane, into the mitochondrion, across the inner membrane and into the mitochondrial matrix. I'm going to draw some arrows on here, recognizing that yes, this is a symport where we're getting the simultaneous influx of a proton ion driving the influx of pyruvate. Those of you who are really dorking out right now are going to say to me that this is a great example of a secondary active transporter. That is we recognize that this is drawing on the energy of a gradient. Specifically, the gradient here is the proton motive force. So pyruvate is like, yeah, dudes, we're riding the PMF as it comes into the matrix of the mitochondrion. Now, this particular transporter has a name. It is pyruvate translocase, that secondary active transporter that gets pyruvate in. So now, at this point, once pyruvate has made its way into the mitochondrial matrix, now is the time when further oxidation goes to town. And we're going to look at the nuances now of the stripping or ripping of more high energy electrons from pyruvate.